This is video number 2 in the blood series, and here are our learning objectives. Number 1, we want to look at blood cells, all blood cells. Just to be clear, remember that in a previous lecture we talked about the presence of red blood cells and then the presence of white blood cells as well. White blood cells we said were immune cells, as well as these kind of things that are called platelets. And we said they're not really straight from a cell because they're exploded tidbits of a larger cell called a megakaryocyte. So in any case, how are all of these things born? How are platelets born? How are white blood cells born? How are erythrocytes born? And that's simply through a process, these processes that are listed here. And so erythropoiesis is a fancy name for making red blood cells. Leukopoiesis is a fancy name for making white blood cells, the immune cells. And then thrombopoiesis is a fancy name for the process of making platelets. Now, platelets go by another name, and that other name is thrombocytes, which is why that term thrombopoiesis there. Okay, so these are just names for the process of creating these cells in your body. All of those fall under this umbrella term called hemopoiesis, also hematopoiesis. These are the same thing, potato, potato. And so all that means is hemopoiesis is simply the process of making blood cells, and it can come in one of three different flavors: erythropoiesis, leukopoiesis, or thrombopoiesis. All right. And where our blood cells derive from, we will get into that. All right, so this is a picture. You got white blood cells there. You can see clearly platelets and red blood cells. That's nice. But if we look at this kind of this blood smear here, we're seeing that all these, basically the bulk of what you see on your screen right now are red blood cells. These are all erythrocytes, again, which are red blood cells. These big purple blobs, because of the stain, these actually are white blood cells. And here it shows you a white blood cell. Well, there's a neutrophil right over there. Here's a white blood cell. That's a monocyte. So these are different types of white blood cells, but they're white blood cells nonetheless. And if I didn't mention to you it before, then leukocytes equal white blood cells, okay? And that's why that name leukopoiesis is there. So leukocytes, those are the same thing. That's just a fancy name for saying white blood cells, all right? So now let's look at this. What we have here at the top is this thing called a pluripotent stem cell. You've probably heard of stem cells at some point. I think you've covered in previous lectures or semesters. Stem cells are relatively undifferentiated cells. There's different levels of stem cells, and we don't really need to get into it, but there are cells that are completely undifferentiated, and then there are cells that are partly undifferentiated. If you don't really know what that means, it means this. I want you to picture, let's say, I don't know, a freshman in high school, and you've got, a ninth grader in high school, and that ninth grader, the sky's the limit. That ninth grader doesn't know, do I want to be a police officer? Do I want to be a lawyer? Do I want to be a small business owner? Do I want to be an accountant? Do I want to be a nurse? Do I want to be a physical therapist, an optometrist? Do I want to be a geologist? The sky's the limit. It can be anything. There are so many potential fates for this particular person. So pluripotent means plural. Potent in this case means potential. So that ninth grader is a pluripotent stem cell, can become anything. On the other hand, there are other stem cells, and now let's take, let's say in an ideal setting, maybe a second-year college student. A second-year college student at that point more than likely has realized, for example, like, oh man, I still don't know exactly what I want to do, but I know for dang sure I am not going to be an accountant. Like that I know for sure. I know I'm going to be something in the health field, like whether I'm a nurse, a doctor, a physician assistant, optometrist, that I haven't figured out yet, but I know it's the health field. On the other hand, other people might say, I know for dang sure I'm done with science. I'm done with any kind of STEM. I want to go into accounting. I'll just pick accounting. I'm going to go into accounting, but maybe I don't want to do accounting. Maybe I want to do marketing. Maybe I want to do finance, something business oriented. And so these are partially differentiated people, partially differentiated stem cells. And we're seeing both those levels here. This pluripotent stem cell can become anything. And then at one step of differentiation, some of them become these myeloid stem cells and others become lymphoid stem cells. Now, personally, it's not so important to me that you memorize, okay, which cells derive from the myeloid stem cell line and which cells derive from the lymphoid stem cell line. It's nice information to know, and in a later course, maybe you'll be asked to actually, remember that in particular. But all I'm trying to show you guys here is that there are different, there's the step of partial differentiation that once you now become a myeloid stem cell, at that point, you've now restricted the number of possible outcomes you can be. You are going to be either a doctor or a nurse or a physician assistant or a physical therapist or an optometrist or a whatever. On the other hand, if you're going to be a stem cell line, you can still become a number of different things. You can become, an accountant or a person in finance or a director of marketing or something like that, kind of in that field. And so what I want you to see then is that these blood cells in the myeloid stem cell line and these blood cells in lymphoid stem cell line, all of these have really a common mama or papa, however you want to think of it. And they're really kind of the same family. Just like we would kind of say, again, I'm going to beat the same drum here, nursing and occupational therapy and physical therapy and medicine, it's kind of all in the same family of things. Whereas accounting and marketing and finance are kind of all in the same family of things. There's greater similarity between those fields than there are in other fields. And so anyway, this is kind of how these various blood cells are derived. Now, what I need you to see is that here is your red blood cell, and this really is where we're going to spend most of our time. And this thing here are the platelets or the thrombocytes. And as I've mentioned to you guys already, these platelets are derived from this big monster cell called a megakaryocyte. I mean, the name itself sounds impressive. It's a megakaryocyte. So megakaryocytes eventually blow up to form platelets. And this process right over here, this birth of a red blood cell, we'll have another slide down the road where we'll take a slightly closer peek at that. And any the rest of the stuff, the details, personally for my students isn't that important. I just need you to understand, all these blood cells are derived from these pluripotent stem cells. There is a step of differentiation which helps determine which stem cell then you will become. Okay, so let's go ahead and zoom in on the red blood cells. What I want you to see first are, let's look at some properties of red blood cells. So we'll list these out and spend a little bit of time talking about them. So red blood cells, the shape of it is a biconcave disc. So what does that mean in English? It just means, like, imagine you had a jelly donut. And if you had that jelly donut and took your finger and you not poked a hole in it, but depressed the top center of it, and then flipped it over and depressed the bottom of it. That's what leads to this kind of shape. You can see how there's an indentation. This is that jelly donut split in half, and somebody poked their finger right up there, and on the other side did the same thing. This is a biconcave disc. So why do we have the biconcave disc shape? Well, the thinking is that when you do that, you will actually increase the surface area, which is going to increase the rate of diffusion. We can talk more about that later, but what I do want to say is that when you increase the rate of diffusion, you increase the opportunity to dump off oxygen, the rate at which you dump off oxygen. Okay? If I didn't say it before, what is the function of red blood cells? It's to carry oxygen. That's its main thing. It picks up oxygen at the lungs and then dumps them off at the tissues. All right. Number 2, red blood cells have no nucleus or mitochondria. So what's up with that? This is weird, right? Every cell that y'all have seen so far have a nucleus, and they've got mitochondria in there. Mitochondria, remember, is the organelle that makes ATP. And so, well, why is that? And the reason is this. The reason is that red blood cells are the guys that are going to carry oxygen. And so we don't want to take a risk that, let's say, en route from the heart to the big toe, all right? I don't know. Let's just say red blood cells pick up oxygen at the lungs, they go back to the heart, and then they get shot off to the big toe. So we don't want to take the risk that on the road, on the way to the big toe, red blood cell is going to gobble up all that oxygen. Okay, so we get rid of mitochondria to make sure you are not sucking up all that oxygen, or you're not eating that oxygen en route. And so we don't have mitochondria. If that's the case, how do they make ATP? Glycolysis. They've got glycolytic enzymes in there. Okay. It's got a flexible cytoskeleton. We can look at this a little bit more later, but red blood cells travel through blood vessels, and some of those blood vessels get really, really narrow. There are roads that are really big, picture like an 8-lane highway, for example, and then there are roads that are really, really small. Picture like navigating in a city between two buildings, and it's really, really quite narrow in there. You'll never get like a large pickup truck through, but you can squeeze through, like, I don't know, a Mini Cooper or something. And so red blood cells often have to squeeze their way through, and as they squeeze their way through, sometimes they get bent out of shape a bit. But because the cytoskeleton is flexible, it works out. Now, what is a red blood cell? To be honest with you, it really just is nothing but a big old bag of hemoglobin. Okay, there's no nucleus, there's no mitochondria in there. It's just a big bag, a big sack of hemoglobin. And hemoglobin is a protein, which we'll look at in a bit. And then the next property of red blood cells you need to know is that, they don't live very long, 120 days. If you don't have a nucleus in there, then you're not really doing a constant protein synthesis, which is going to permit regeneration of different things. So if you can't do that, then you're going to have a short lifespan. So these guys live for 120 days. They're born in the bone marrow. Bone marrow you guys saw when you looked at bones, and that process again is called erythropoiesis. And eventually, at around day 120, they die, and they die in the spleen or liver or bone marrow. But just to keep it simple, we can say they die in the spleen. And we'll have a separate video that looks at kind of the death and recycling of red blood cells. What I want to do now is zoom in on hemoglobin. So if we zoom in on hemoglobin, here's what we're looking at. Hemoglobin is a protein. Now, if you remember what you already know about proteins, remember that proteins are made up of these polypeptides that have undergone, regional foldings and secondary structures and then tertiary structures where the whole thing folds on itself. And then remember that some proteins have a quaternary structure, which is when you have two or more polypeptides that do their thing, and then they all hook up together. So what we're seeing here is hemoglobin is actually a protein with 4 different polypeptide chains. Now, 2 of them are what we call beta chains, 2 are called alpha chains. What's the difference between beta chains and alpha chains? For us, honestly, it doesn't matter. Who cares? Anyway, they're 2 polypeptide chains that are called beta chains, 2 are called alpha chains. And what you're seeing, these guys are pictured here. Now, this particular artist is drawing the dark ones in here as the beta chains and the light ones in here as the alpha chains. For all intents and purposes, for us, there's no meaningful difference for our purposes. Now, within each of these chains is a flying saucer. Now, if you look where my cursor's at right over here, it looks like there's a flying saucer with that red in the middle and then that disc-like yellow thing. There's another flying saucer, there's another flying saucer, there's another flying saucer. Now, if we blow up that flying saucer, or maybe not blow up, that's probably a bad use of the term. If we zoom in, I should say, on the flying saucer, here's what we see. This flying saucer is called a heme group. Heme. This is why this is called a hemoglobin molecule. Hemoglobin molecule. Okay? And so this thing is called hemoglobin. So if we look at a hemoglobin molecule, what is there? I mean, well, like I said, there's a red dot and then the yellow disc. The red dot in this depiction is an iron ion. The flying saucer, the yellow disc, is something that we call a porphyrin ring. And now if you look at that porphyrin ring, you'll see how all these crazy covalent bonds between carbon and hydrogen and nitrogen are there. And that's nice and good. We don't need to know the chemical details of the porphyrin ring other than to recognize that it's this part right over here, where right at the very center is iron. Now the presence of that iron is really, really important. Okay? Now, why do we have this at all? What's the point? So remember, let's just kind of get our bearings again. We're in a red blood cell. What is a red blood cell? It's just a big old bag of hemoglobin. Inside, you've got millions of molecules of hemoglobin on the inside of a single red blood cell. And then so there we cut open the red blood cell and we see millions and millions of this entire structure right over here. Now, each of those structures, again, has 4 polypeptide chains, and each polypeptide chain is associated with a heme group. Now, what is a heme group? The yellow disc and the red dot. The yellow disc is the porphyrin ring. The red dot is the iron ion. Now, this brings us to the question, okay, that's great. So I've got tons and tons of these heme groups all over the place with iron in the center. Why do we care? And the reason we care is oxygen. When oxygen shows up, I shouldn't say it shows up. When the red blood cell gets its way to the lungs, and then oxygen is brought in. Oxygen binds right over here. So if I were to just make, I'm just going to call this oxygen. Okay, so there's O2 right over there. And so now we've got the oxygen molecule, and I put it in here as O2 because it's actually 2 oxygen atoms covalently bound to one another. And so this is your molecular oxygen that you just breathe in, and boom, there it goes. Oxygen is now comfortably and happily seated right on top of, or I should say right within that heme group. So this is why you need the heme group. Now, here's the thing. If you don't have heme groups, oxygen has nowhere to sit. These proteins cannot carry oxygen. They just can't do it. It's actually the heme groups that carry oxygen, and then the rest of the protein, the polypeptide chains, carry the heme group. Okay, so a bit of a chain there. The other thing about it is this: if you don't have iron, for example, maybe you have nice little heme groups here, but you don't have that red dot in the center. You don't have iron. Well, now you can't carry oxygen anymore. But that iron is a really, really critical part of it. And so anyway, both are important. That porphyrin ring as well as that iron right in the center, both of those are really important because you want to carry oxygen. Now, I want to mention this now. For those of you guys, well, all of you at some point should study the respiratory system. So I'm going to mention this now. I'm going to burn this into your brain. Why does oxygen need to be seated here? Why do we need a protein to carry oxygen around in the blood? Well, if you've already covered the endocrine system, then you should know, look, we've got proteins that carry things that are not water soluble. If things are water soluble, they float through the blood easy peasy, no problem at all. But in this case, what this tells you then is that, yeah, oxygen is not very soluble in water, in blood in our case, right? But blood is mostly water. So oxygen is not very soluble in blood. And that's why we need these protein boats. It's just that now these protein boats are actually loaded inside these red blood cells. And that's something that we haven't seen before. Okay. All right, let's talk about then anemia. What is anemia? Anemia is simply a reduction in oxygen carrying capacity. I'm going to write that out here. So there it is. Anemia is a reduction in oxygen carrying capacity. And so, let's just to make up numbers here to make life simple. Let's say normally your blood can carry 1 million units of oxygen at any given point in time. And so if all of a sudden, instead of being able to carry 1 million, you're now reduced to 70% capacity to 700,000 units of oxygen, well, now you have anemia. You have a reduction in oxygen carrying capacity. Now, here's what that leads to. A lot of times when people say anemia, we instantly think iron deficiency. Well, just to be clear, iron deficiency is only one type of anemia. In other words, how can you mess up oxygen carrying capacity of the blood? There's a lot of different ways. You can blow up red blood cells. You can deform red blood cells. You can make red blood cells really, 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 really small. You can make them really big and ugly so that they don't actually work very well. You can just eliminate it of iron. You can get rid of the proteins. My point is there's a lot of different ways you can mess up with oxygen carrying capacity. And so it's just that a reduction in iron is probably one of the most famous ways out there, but they do not mean the same thing. Iron deficiency anemia is a type of anemia. It is not the same thing as anemia. It's just one type. So another type of anemia is this thing called sickle cell anemia. So what happens in sickle cell anemia? In sickle cell anemia, these polypeptide chains, there is an error in one of those amino acids. And so when there's an error in one of these amino acids, then the protein itself is just a little bit different, right? Because form and function are married together. So in the protein itself, the polypeptide chains are just a little bit different, then they work differently. So what ends up happening is that when oxygen binds to the heme group, then there's actually kind of a weird shape, a little bit of a shift in the conformation or the shape of hemoglobin. And when that happens in mass, then you start to see a kind of a twisting and a distorting and a bending of the entire red blood cell. Because remember, red blood cells are nothing but big bags of hemoglobin. And so when that happens, that causes this change in shape. And you see here, hemoglobin molecules line up and form chains. They do this weird behavior, and that changes the shape here. And so these sickled erythrocytes, again, it's just not good. We don't like sickle cell anemia. It changes the way they shape, therefore it changes the way they function in their ability to deliver oxygen in an optimal way. They can end up actually plugging up small blood vessels. Remember I said that some blood vessels get real small, like a narrow alleyway in a city. And so if you have a nice, healthy, I'll say plump, maybe plump's the wrong word, but supple and flexible red blood cell, then it can sneak through those parts. But once you deform it like this, you're not quite as pliable and supple as you were before either. So you're more likely to get stuck in small blood vessels, or even pop once you get into them. All right, so we don't like sickle cell anemia. Anyway, let's wrap it up. We talked about where blood cells come from, and we defined these different terms out there. We looked at general properties of a red blood cell. We looked at what hemoglobin looks like and why iron is important, as well as defining anemia and sickle cell anemia. And that will wrap it up for lecture number 2.